Thank you. Good afternoon, everyone. Happy New Year, and welcome to our EGFR and KRAS Targeted Precision Therapy panel. I'm Kaveri Pohlman. I'm a Senior Equity Research Analyst at Clear Street. As the name suggests, this panel focuses on the KRAS or RAS and EGFR targeting drug space, which has become very exciting. The focus of today's panel is going to be on some recent developments in this space. What's the potential of this area, and what are the opportunities, and where are those opportunities? Both KRAS and EGFR spaces or the markets have meaningfully grown, and that's because these targets are vital or important for multiple tumor types, for example, lung cancer, colorectal cancer, pancreatic cancer, but they are also very tricky to target. That's mostly because these proteins are also involved crucially in normal development process. So some of the earlier approaches, with narrow therapeutic index, they reinforced the undruggable view for these targets. And it was not till 2003, 2004 when we started seeing drugs in the market for EGFR with approval of gefitinib and cetuximab. For the EGFR market, it has grown significantly. Specifically, the EGFR mutant market right currently is actually more than $15 billion. And it is expected to be more than $30 billion by 2032. There's a lot of M&A and licensing activities in this space. J&J's amivantamab, which is already in the market, it's an approved drug, that was designed by Genmab. And we also saw a recent acquisition of Merus for $8 billion by Genmab. And it makes sense because, there's a lot of interest since we have seen a lot of success with Tagrisso, which is also called osimertinib, and amivantamab. And we also have some exciting drugs coming in the EGFR space with 2 TKIs that could get approved next year from Aravent Bio and Cullinan Therapeutics. Similarly, I would say KRAS space, it has exploded after sotorasib's approval as the first KRAS inhibitor in 2021. And, the M&A activity here is also very strong. AstraZeneca recently did a deal with Chacko Bio. And it seems like Revolution Medicine, which alone has more than $20 billion market cap, seems like it's in discussion for acquisition. And, the KRAS space, it's currently a smaller market, but it is growing very rapidly and with development of even pan-RAS inhibitors and degraders. So what differentiates these drugs? What makes them really attractive? What is special about them? How are they better? What is the potential of these drugs, these new assets? And with all these efforts going on in the space, where are the opportunities? That's the focus of this panel. And to have this discussion today, we are very fortunate to have David Chacko from Erasca, Dozie and Amuzie from Johnson & Johnson's, Duncan Young from AstraZeneca, and Stefanie Flückiger, from TOLREMO Therapeutics. Thank you for joining us today. So maybe just to start things off, if you could give us an overview, tell us about yourself and give us an overview of the technology platform you have, what's differentiating about it. And from the big pharma perspective, I would also ask David and Dozie and Duncan to comment on, some of the market progress you have seen with the approved drugs.
Hi, everyone. I'm David Chacko. Good to be here, and thank you for the invite to come. I'm CFO and CBO with Erasca. We're a San Diego biotech company. Our name is our mission to erase cancer, and there's a double meaning in that name as well, to eradicate RAS-driven cancers. And so the topic of this panel today is, quite on point for what it is that we're doing. We, as a company, have been around since about 2018, and in that time, we've explored various parts of the RAS-MAP kinase pathway. We now have 2 RAS programs that we call ERAS-0015, which is a pan-RAS molecular glue. For those who are familiar, as Kaveri mentioned, with Revolution Medicine, this molecule, 0015, works through the same mechanism of action as RMC-6236. And we also have ERAS-4001, which is a pan-KRAS inhibitor. Both of these programs are in their phase 1 dose escalation. We've publicly guided that we will share clinical data on both of these programs this calendar year. And we're really excited about both of them. I know we'll get into that in the course of this panel. But we do think that, as you mentioned, the RAS space is relatively nascent in terms of sotorasib and adagrasib with their approvals. I think there's a lot of other areas that can be approved or explored and then potentially have drugs approved in those spaces, and we're looking forward to that very much.
Thanks, David. I'm Dozie Amuzie. I'm from Johnson & Johnson, from Oncology External Innovation. And my role there is to support external innovation opportunities for oncology in West North America, which is anywhere west of the Mississippi River. I'm a scientist by training. I've been with the company for 10 years in different roles. For relevant to this panel today is amivantamab, which I actually happen to be privileged to be part of the big crew that helped move that along in development when it was EGFR-c-MET bispecific. So one thing that maybe some in the audience may not know is that the way that J&J approaches diseases in general is we have what we call disease area strongholds, which is that the effort is focused on understanding disease biology and then building around usually one major asset and then building around the asset. So for our lung cancer franchise, the foundational asset then is amivantamab, which is EGFR-c-MET bispecific. And it was built differently. It's not just binding to those receptors, as you know, from comparing with things that just bind to EGFR. There's other things around how it engages effectors and things that you could learn about how amivantamab is built. But that has enabled exploring that biology for patients now in lung cancer. Just ASCO GI just concluded, where some data was shared for colorectal as well. And so the thought then is that everywhere where EGFR is playing a role, amivantamab could play there. And the team is trying to understand more in which patient population is it transformational or can it be transformational, but also with what combinations is going to make sense. So amivantamab is the bedrock of our lung cancer franchise, and we're sort of building around that as we could talk maybe in a little bit more detail during the conversation.
Thanks. Good afternoon, everyone. So I'm Duncan Young. I head up the oncology search and evaluation team at AstraZeneca. It's a team of about 7 of us covering all modalities, all geographies, all stages of development between us. I've been with the company on and off for 10 years or so, with a brief stint in biotech before coming back to the BD team 2 years ago. Obviously, AstraZeneca has a long history in the EGFR space, first with Erasca and then more recently with Tagrisso, our TKI efforts in this space. And I think it's important to realize that you don't get to a position where you are with Tagrisso without the long history of Erasca and understanding how you develop drugs against these targets, how you understand mutations, resistance arising against these targets, and develop a next generation molecule to address that. Towards the end of last year, we also announced a proposed partnership with Chacko on their pan-KRAS molecule, and that's going to form part of our key ambition to move forward into gastrointestinal tumors, building on some of the other assets that we have in this space. I think one of the key things, and then to set Stefanie up, is thinking about how it fits with the rest of the portfolio that we're building and how important combinations are going to be to truly address tumors driven by these targets as well.
I am Stefanie. I'm co-founder and CEO of TOLREMO Therapeutics. TOLREMO is a private Swiss-based biotech. We actually don't have any KRAS or EGFR inhibitors. We develop therapies that target non-oncogene addiction. So going after biology that drives tumors in parallel to a driving oncogene. Our lead asset is an epigenetic inhibitor. It's a CBP-P300 bromodomain inhibitor in the clinic. And we've seen that especially in lung cancers that are EGFR or KRAS mutant that do not have a strong addiction to the oncogene. That's where we see the most monotherapy activity of our epigenetic modulator. And our goal is, of course, then combine it with oncogene targeting agents for deeper and longer responses in the clinic.
Great, so a combination partner. Thank you, that's helpful. And maybe I know I did a lot of talking in the beginning, but I would also love to hear from our panelists here what they think about, what their point of view is on these areas, how important those are. From a big pharma point of view, I would like to really ask Duncan and Dozie, how much value these areas have generated for the companies, and and how important they are in terms of companies' development. And to David and Stefanie, I would love to know, why they chose these areas of drug development, and what are some of the exciting factors.
So I think Dozie used the word stronghold. We sort of maybe rival strongholds in the lung cancer space when it comes to EGFR. So obviously Tagrisso is a key asset for us, and importantly one that was homegrown, developed internally, that we're using to drive our leadership position in lung cancer. I think with key targets like EGFR, it's important that you look at different ways to address that target and different ways to address resistance that will likely inevitably come through treatment against that target. So we have multiple different modalities that we are exploring. We have radioconjugates and ADCs targeting EGFR and c-Met, and we have cell therapy approaches that we're looking to address KRAS, as well as small molecule approaches. And I think one of the important things that we're building with our portfolio is all of these different modalities, all of these different approaches to a target, orthogonal mechanisms of cell kill, whether it's directly killing the tumor cell or whether it's engaging the immune system to support that, because you need to hit tumors from multiple different directions to have more durable responses and improve patient outcomes here. So this is a key. Tagrisso is now a multi-billion-dollar opportunity for us and a key part of our lung cancer franchise. I think as we're looking to KRAS, as I mentioned earlier, it's really going to be about how we can build across the key KRAS-driven tumors, so lung, pancreatic, and colorectal cancer. And that speaks to then some other combination strategies that we'll be looking to bring in within agents in the portfolio.
Okay, I'll go after that. I think for us, within lung cancer, what we've been talking about mostly is non-small cell lung cancer with EGFR, where amivantamab is playing. But within the disease area stronghold, we are looking at that asset across the board for head and neck, for where you have the origami studies, and also for colorectal. But we're not stopping with non-small cell lung cancer and these molecularly driven cancers where these mutations play a role. We're also paying attention with other assets to wild type, non-small cell, small cell as well, but also squamous. So we are focusing on the biology, and within that biology then the combinations are driven by understanding the resistance mechanisms and what could combine well to bring more transformation. So if you look at Mariposa and Mariposa 2, those are exploring different kinds of combinations and to see where what kinds of drugs can combine with amivantamab to raise the bar a little bit. So I think the principle there is finding the right target, finding the right monotherapy for the right target within the biology, and then beginning to explore combinations that make sense, paying attention to resistance.
For Erasca, as I mentioned, the company itself, we've been around since 2018, and then we went public in 2021. And at the time that we went public, RAS was a big part of our story as well. We had other assets at the time, but RAS was an important one for us. We had internal KRAS G12C as well as KRAS G12D programs. One of our co-founders was Kevon Shokat here at UCSF. And he was the one, Kaveri, you mentioned sotorasib and adagrasib and the original, set of molecules that led to kind of this explosion of great innovation in the RAS space. A lot of that was the work that Kevon Shokat did to find the switch II pocket on the KRAS protein that allowed it to become a druggable target. And so we were able to capitalize on a lot of his learnings in terms of how we thought about our programs. Now, over time, as the field has evolved and as our thinking has evolved, we've moved on beyond just the mutant-specific programs and brought into more of the pan-RAS and pan-KRAS approaches that I mentioned earlier. We think that these are really exciting. There's a lot of unmet need here, unfortunately. There's a lot of patients that are suffering from these types of cancers, 2.7 million patients around the world that are diagnosed annually with RAS mutant tumors. And so there's a clear unmet need here that still, needs to be fulfilled. And so we felt like these were good ones for us to, really focus on right now. I think both Duncan and Dozie, you guys mentioned just the nature of this pathway is that it's a wily set of targets to go after, that you have to think about combinations, you have to think about where's resistance going to come up, and that's been an important part about how we think about our development, and whether that's combining with our own in-house agents, whether that's combining with best-in-class standard of care agents from external. We're agnostic on that because we just want to find the right combinations for the right patients. And then in terms of modalities, we are also modality agnostic. Right now we have a small molecule pan-KRAS. We have the molecular glue pan-RAS molecule. But in the past, we've actually had a degrader program. We actually have a EGFR bispecific antibody that we call ERAS-12. And then at one point, we had actually made an equity investment into another company called Affinity Therapeutics, which was working on T-cell therapies for KRAS. So that was one that we knew that as a small company, you don't want to just dabble in cell therapy. You're either all in or all out. But by making an equity investment, we were able to be on the front seat of a company that had the likes of Jim Allison and other wonderful members, advancing the KRAS space from the cell therapy standpoint.
For us, the reason why we went into KRAS and EGFR-driven lung was really, of course, driven by the unmet medical need, but also driven by clinical evidence that resistance does limit patient benefit. And when we look at how originally EGFR inhibitors, but also with KRAS inhibitors, how these orthogonal combinations that we did, they mostly focused on MAP kinase signaling, right? You go up and down the MAP kinase signaling pathway, combining KRAS with SHP2 or SOS1 or MEK or MET. But there is really only so much biology you can get out of targeting the MAP kinase signaling pathway. And we all know that tumors aren't just an oncogene. There is so much more to it when it comes to resistance. There is metabolic reprogramming, there is transcriptional reprogramming, lineage switching that we can't explain by genetic changes. And for us, targeting tumor cell plasticity and an epigenetic mechanism, that was key. Where can we really add benefit? And both for EGFR and KRAS, there is clinical evidence that 50% of resistance is actually driven by non-genetic adaptations that we currently can't target in a clinic because we don't have any drug targets that we can go after.
Great. Oh, that's very exciting. So I guess a question about, since we are talking a lot about combinations here, modalities here, I just want to understand, we do have, so now we have approved TKIs, which are small molecule inhibitors, of course, and amivantamab is a bispecific that targets, like Dozie mentioned, it targets EGFR and c-MET, but we also have degraders coming up in this field, and as well as even vaccines. There are KRAS vaccines being developed. Therapeutics is one of the companies in that space. And besides that, Duncan mentioned that they are radioconjugates, ADCs, as well as cell therapies. So I would like to maybe kind of dig a deeper into this area of how they think about these modalities emerging, whether, like you said, starting with monotherapy and once resistance develops, going in combination, and even in those areas, what kind of combinations make sense, and what modalities do you think succeeding further, or it's more going to be like a precision medicine where different patient scenarios could lead to different combination approaches?
I can start there. I think the way this works at J&J within the disease area stronghold is you have the asset we have the combinations we have, and we are learning continuously. And it's the deep biology within those learnings, the biomarkers that emerge, the insights around resistance and the responders and non-responders that begin to raise hypotheses around which combinations would make sense. And this is true not just in lung cancer, it is true with myeloma, where you just saw the Majestic-3 data released. Those combinations are driven by insights that come from clinical trials that begin to suggest, okay, if this is what we think about the resistant mechanism, these combinations would make sense as a hypothesis. So we let the biology lead where we go. And then because of that, we are truly modality agnostic, that it would choose the target and it would choose the form of the targeting that would make sense. So I think that's the key in terms of how we approach it. We keep an open mind around which assets to combine, provided that is going to allow us to extend benefit to more patients.
I think, as Stefanie said, that there's been an incredible amount of work that's been done with the first KRAS inhibitors that went into the clinic, sotorasib and adagrasib, and looking at these mechanisms of resistance. That the one thing we know inherently that KRAS and the MAPK pathway can do is mutate and adapt, and that's where you see relapse coming from. So, but you see, as Stefanie was saying, you see on-target resistance mechanisms, you also see off-target resistance mechanisms. So we very much think about that when we're building out the portfolio. Oncology is now about combination regimens, and what we do is look at how a patient may present, what stage they may present, and how you have the right options within the portfolio to address that. So you might be looking to have an ADC, for example, to come in and debulk the tumor. You may be then looking to come up with a small molecule approach. You may be looking to come up with a cell therapy approach or an IO approach to address what remains of the tumor. Then you might be dealing with micrometastatic disease, and you could mop that up with something like a radioconjugate. So very much how you have the tools to build that regimen and put those pieces together is key. And underpinning that is something that we were doing a lot last year, which is from a BD perspective, looking at how we bring in AI models, and the partnership that we announced with Tempus and Pathos to build the world's largest oncology foundation model will really help us optimize how you combine, how you sequence, and how you bring the right options to these patients. You can monitor that now way better with ctDNA approaches, for example, than you could do previously.
No, I think just a question of sequencing will be very important too, right? We've seen it with FLAURA2, where you move chemo back up in the first line. Is that what you want to continue doing? Is that what patients want, right? And what comes after that?
Great. And then I guess another question about, we do have, like I said previously, these targets were considered undruggable, but I would also like to kind of understand a little bit about, what has been addressed so far and what hurdles we have overcome with the ongoing efforts, what are the key challenges that we still see that remain in this space?
Yeah, I'm happy to start. I mean, I think the progress that has been made is fantastic in both the EGFR and the KRAS space. And, again, just given the amount of time that these targets have had approved therapies, and especially in the KRAS space, that it's been, just such a short amount of time, we've learned so much. But that said, and actually let me talk first about KRAS for a little bit. The fact that it was historically an undruggable target for so long until Kevon Shokat and his team identified that switch 2 pocket that I mentioned earlier. This was a very tough target to go after. And his paper in 2013 that allowed for this first wave of compounds, that led to an explosion of ideas around where to go. And what has come up, though, is that there's been various challenges of various sorts, whether that's adaptive resistance that comes up, whether that's the challenge of combinability that was seen with some of the initial inhibitors, particularly with CPIs, or whether it's tolerability more broadly. And so I think that there is still tremendous progress that has been made, but a lot more still to go. You see that in terms of response rates that look, again, relatively decent in terms of the fact that this was a historically undruggable target, but still room to improve. And I think that's one of the things that's very exciting, at least to me and to us at Erasca, that you do see glimmers of promise. You see these green shoots of promise where, you see, for instance, in a particular tumor type, you might see a 30% response rate from one drug, but maybe you start to see a 40% if you from a different drug, and maybe it goes up to 50% if you start thinking about combinations. And all of those are just nice steps that are being made on top of the what's been coming before. But obviously, there's still a lot of headroom for improvement. Why can't we get from that 30 to 50% to something higher than that? And so I think that there's a lot of room for improvement. In terms of where these drugs currently are. And I think that's where combinations can come in, and I'm just talking about one point there in terms of response rate. Now, you also have to think about durability of the response. You also have to think about what the AE profile looks like and what is it that patients are able to withstand. And so I think we as a field, owe it to patients to identify, these new emerging opportunities to combine, to look for better ways for us to treat them.
I mean, what I think is David's saying, it's easy to sort of take for granted just how quickly things have moved in the KRAS space, and now we're not just talking about cracking it; we're talking about cracking other specific alleles and how quickly that world has moved on. But yet there's a lot, though, with those first-generation G12C inhibitors that they paved the way for cracking KRAS as a target. But then there were liabilities with them, the tolerability in combination with checkpoint inhibition. But also, if you look at the PK profile of these molecules, it's very hard to get the full coverage on the target, sustained inhibition of the target and the pathway. With that sort of PK profile, and that's where you see the next generation molecules coming on. So I think that there's the combination element, but there's also thinking about what a true best-in-class profile looks like against some of these targets, be it KRAS or EGFR, in terms of the ability to hit the target and get sustained inhibition of the target in the pathway, but also maybe address arising resistance, adaptability on the target. And so we also think about this a lot. There was a comment in one of the earlier panels talking about it's not a first-in-class world anymore, it's a best-in-class world. And Susan Galbraith, our head of oncology R&D, has also talked about the fact that what keeps her awake at night is not loss of exclusivity on the portfolio, it's loss of relevance, it's how quickly oncology innovation is moving forward against these and how quickly new molecules are coming through that are able to offer improvements on profiles, maybe before that first generation has fully worked its way through clinical development to get on the market.
And I think there too, right, there is probably how much additional efficacy can we get out based with better molecules? And is there a threshold that we hit? If you have the perfect molecule, would you get 100% CRs? Probably not, right? Because tumors are just too dynamic and too complex, really. So there the question is, what are the best combinations and which mechanisms of action can we really get into the clinic with a good tolerability profile so patients can actually stay on these drugs for a long time, which becomes more and more important the longer they stay on, right?
And combinability is a key aspect of that. So having the right molecular profiles to allow for that kind of combinability, absolutely.
Yeah, I'd add that the best-in-class idea resonates, and with EGFR, I think there's still space to innovate around how do you truly target in a cancer-specific way and totally avoid normal tissues where EGFR is expressed in skin and gastrointestinal tract. So innovation is emerging in that area, and it's conceivable that there will be even further generations of EGFR therapies that would be a little bit better at avoiding normal tissue.
I mean, that's the thing when we think about some of the biologic approaches, and the toxicities that you associate with hitting EGFR in the skin, for example, and can masking approaches, can conditionality, either conditionality within the tumor microenvironment or some form of logic gating help address that. I think that's where it's quite an interesting, exciting space as we're seeing innovation come through in biologic engineering to support that.
Well, I also think, in oncology, especially compared to maybe other therapeutic areas, just the balance that you need to find between safety and efficacy, right? And those are kind of a push and pull sometimes. I mean, so I'm thinking, for instance, in the RAS space, there has been a hypothesis around the fact that a pan-RAS molecule that hits all three isoforms of RAS, meaning H, K, and NRAS, may have a worse rash profile because it hits H and N as well as K, versus a pan-KRAS that spares H and NRAS may have a better rash profile. Now, I think if you look at just two pieces of information that have come out earlier this year on that, this talks about kind of the push and pull. So on the one hand, there was data earlier this week from BridgeBio Oncology where they showed their AE profile, and they're a pan-KRAS molecule, that they don't have much rash. And so that actually does support this idea that if you spare H and NRAS, that you could actually have a better rash profile. Now, on the other hand, the second piece of information that came out earlier this year as well is a paper from C. Lover et al. That talks about the resistance mechanisms that may come up, and that if you spare H and NRAS, that there could be bypass via PI3K, and so that a pan-RAS may allow you to actually prevent that pathway for resistance. And so it's, again, this kind of push and pull between safety and efficacy where, yes, on the one hand, pan-RAS may cause rash, but a pan-RAS may also prevent adaptive resistance.
Or it could be using degraders instead of inhibitors that can get rid of the issue. All right, so more specificity leading to better safety that can allow combinability. Great. Oh, thanks. And I guess one of the areas is huge, lung cancer space. It's a huge market, but would love to understand, where are the market expansion opportunities beyond lung cancer? Where do you see, I know this is Dozie's favorite question, so, but would love to know, if there are any other proof of concept data. I know you mentioned that you're testing amivantamab perhaps in colorectal, so maybe Dozie can start with.
For full disclosure, I told Kaveri I don't want to talk about market opportunities, but we follow the science wherever it leads for benefit for patients. And what has emerged is obviously EGFR has a role in colorectal cancer. It has a role in head and neck. So with the asset we have, there are ongoing studies to explore whether this would be beneficial for patients in those spaces. But I think it highlights a concept, at least, of how I think maybe other companies do this too. But at J&J, it's really to let the science lead us to provide benefit for patients. And that's quite rigorous, because even when we go to head and neck, it's not head and neck for everybody. We're asking questions around which segment of head and neck would this be most useful. And I think it's those kinds of questions that that drive then with the kinds of clinical trials that are designed, if combinations are necessary initially, or if combinations is something you bring later on. So for amivantamab, yes, the origami studies, as I mentioned before, are going on both in head and neck and in colorectal, and we'll see. We're hoping to see some data come out from there and lead to expansion opportunities.
I think one of the interesting things to think is how new modalities and new approaches and improvements on these molecules will allow and unlock some targets in particular indications. So we know that cetuximab is part of standard of care in colorectal, and if you're thinking about taking KRAS molecule into that setting, you're going to have to think about combinability with cetuximab, and speaks to the rash issue that David was talking about. But then when there are companies out there that are taking KRAS in molecules into pancreatic that are then looking at combinations with cetuximab in that setting, in the pancreatic setting as well, because ultimately it's part of that downstream signaling pathway from EGFR. So I think we're quite interested in how new approaches can unlock some of these targets in other indications as well. And that might speak to particular modalities, to particular different approaches where you're matching either an ADC payload or a radioconjugate approach to sensitivities in the underlying tumor setting that maybe changes your perspective on how you're using targeted modalities against KRAS or against EGFR in these different settings as well.
I think what is key here, what is interesting, is that lineage really does matter, right? When we think of CRC or head and neck, it's kind of this, we've seen it with BRAF inhibitors in CRC that don't work without the EGFR component. We've then seen it with KRAS inhibitor in CRC that don't work with the EGFR component. And they're just for us to keep in mind that lineage does matter, and tumor agnostic approaches, they might sound very attractive, but sometimes biology just doesn't work the way we'd like it to. Right?
Yeah, I mean, I think on the KRAS side, the KRAS is implicated in so many different tumor types. I mean, Kaveri, in your opening remarks, you talked about the big three tumor types of, pancreatic, CRC, and non-small cell lung. But there's also a long tail of opportunities that are still, sizable patient populations. You're talking about, tens of thousands of patients in the U.S. that are diagnosed with some of those, tumors. And so these are, unfortunately, as I mentioned in some of my earlier remarks, just a huge unmet need that really does need to be addressed, not only in those, big three opportunities, but even some of those longer tail opportunities. And so, there's just lots of different areas that KRAS is implicated in, and where to some of the comments that have been made throughout the course of the panel, where does the biology take us? And we'll see as we continue to advance as a field in terms of where that goes.
And Kaveri, you mentioned degraders earlier as well. So obviously, I think this is another fascinating part. When you think about going against these targets, is there additional efficacy to get from a degrader approach? But then, I think as David was saying earlier, what then are you bringing in? What toxicity risks might you bring with that as well? And ultimately, we need to get into the clinic and find out. We announced a collaboration with Pine Tree Therapeutics, I think it was last year, on an EGFR degrader biologic approach. And again, it's just what can we do to maybe find different ways of tackling these targets and ultimately take it into the clinic and find out.
Got it. That's very helpful. And maybe a question for Duncan and Dozie. Maybe if you can just tell us, how big pharma companies like J&J and AstraZeneca, can partner with mid-cap companies to, expand work in these areas.
Maybe I'll start. I think we start on the premise that the number of smart people outside our company is much bigger than the one inside. So, and a good idea can come from anywhere. Then we look for opportunities that allow us to bring 1 + 1, and it will be greater than 2. So there really has to be a belief that there's something the company is bringing uniquely, and there's something we're bringing from our clinical experience or within our portfolio that would make that union or collaboration quite strong. And within the disease area stronghold format, we are really looking for very differentiated ideas that would come in and further enable or amplify or catalyze what we're doing already. So we keep an open mind. We listen to all kinds of ideas, but then we take a lens of how is it going to help us extend what we're doing now. So it really has to fit within the current. We have to see a path that it merges with something we're doing currently and becomes an enabler.
I think that's a key point. One of the benefits of being a key oncology company is that we've got quite a broad portfolio in oncology, but that then also does bring questions about, well, how do we position, how do we think about new opportunities coming in that fit within the portfolio that we're not doing already, where there's a white space and we're not cannibalizing something that we have already. We are very open to partnering and collaboration and early discussions in this space. I was just quickly making a note down. So I think there's 5 things. If I think about EGFR and KRAS between them that we've looked at, where there are BD deals that we've done in recent years. We partnered with Alorian on an EGFR L858R program, with Usinova on a KRAS G12D, with Neogene, a lot of their TCR approaches that we brought in for the cell therapy franchise are also applicable to addressing KRAS mutant specific targets. Pine Tree Therapeutics that I mentioned, the EGFR degrader biologic, and then most recently with Chacko Bio on the pan-KRAS molecule. BD, obviously I've got a vested interest in saying this, but BD is a key part of delivering on our oncology strategy, and we through the continual scouting and presence, we sort of are able to bring these opportunities to the oncology leadership, where we can find a way of positioning them strategically within our portfolio. So generally very happy to have early discussions. I think it helps where we are embedded within the oncology R&D organization, so we're very tapped into our oncology strategy and able to think about that positioning question from an early stage, and explore either licensing or collaboration opportunities, because the other thing we can do is look at drug supply collaborations. We have platform studies where we can maybe bring in a partner's molecule to look at combinations within the clinic ourselves and find ways of generating the data together.
Sorry, one thing I didn't say, because I see quite a few of my JJDC colleagues in the audience, is that we have JJDC, which is the oldest corporate venture, certainly in pharma. But what JJDC enables is sometimes it's an idea that we really like. It's very good. It seems like it can fit into the portfolio, but we're not quite sure how yet. And sometimes can partner with JJDC to make an investment and leave the idea alone, let it mature on its own. And sometimes it matures to a point where oftentimes it matures to a point where we understand and the company understands. So it's a way to say we really wait for the win-win time. We don't have to, if the science is differentiated, the team is spectacular, there are things we can do to stay in touch and stay engaged. That does not mean research collaboration immediately.
That was very helpful. And I guess we discussed during the panel about how critical safety is, and especially for combinability, which is where oncology is moving. From that point of view, I want to ask David and Stefanie about this. So Stefanie, with your novel approaches, and David with targeting pan, and Duncan, you also mentioned Chacko Bio with pan-RAS. So I just want to understand, with these novel approaches and with targeting something like wild type forms as well, how you think about safety, long-term safety specifically, because these are relatively novel approaches. We haven't established that, and how much risk does it add?
So for us, since we're really a drug resistance company, safety has always been key. And I think that's probably also the approach of targeting complementary mechanisms, that you don't necessarily have an overlapping tox profile, right? So not targeting the MAP kinase signaling at 2 nodes, but really doing something very different in terms of also the safety that you'll get. And so for us, as an epigenetic modulator, a lot of people have sort of this knee-jerk reaction to, oh, that's going to be very toxic, because HDAC inhibitors were toxic and BET inhibitors were toxic and lots of other epigenetic mechanisms were toxic. So I think the next generation of epigenetic molecules, if you have the right target and if you target the right domain, you can actually have very well tolerated epigenetic inhibitors, in case ours sort of doesn't have any overlapping tox with sort of EGFR or KRAS inhibitors. And that's really been key from the beginning of our development, that we would have a very combinable agent.
Yeah, I mean, I think, as Stefanie was saying, I mean, I think safety is paramount. You know, you always have to think about that. It's absolutely very important. I mean, I think that it's a very important consideration in light of oncology and what I mentioned earlier in some of my comments about the push and pull between safety and efficacy. You brought up, Kaveri, specifically around wild type. You know, both of our molecules, our pan-RAS and our pan-KRAS molecule, they actually do hit wild type. And so that could actually has the potential risk of introducing some toxicity because you're hitting wild type, the wild type protein. But we think that that's actually really important to hit wild type because of the potential for wild type active amplification as a resistance mechanism. And we were talking about this before the panel as well, that is an area for resistance and for, you know, even if you see good response rates initially, are you getting durable responses? And so we think that hitting wild type is actually a really important aspect. And so it's about finding that balance on the safety side between, you know, what can the patient tolerate with the efficacy in terms of having a durable response. So that's one aspect. I think another aspect, we talked a lot on this panel about combinations and the importance of those. You know, if you think about the KRAS space, some of the initial players, you know, when they showed combination data with CPIs, with checkpoint inhibitors, you know, I mentioned it in one of my earlier comments, Duncan as well, that was an area that maybe wasn't so combinable. You saw increases in the liver function enzymes and other sorts of issues. I think with some of the more recent data sets that have come out in the KRAS space, the combinability with checkpoint inhibitors look pretty good, actually. And so I think we've made advancements there, which, you know, have to do more with the chemistry of, you know, kind of those earlier generations versus some of the later generations. But I think that that's an area where we have improved what the safety and tolerability profile looks like in combination. The other area too, I mean, we've talked in some of my previous comments, I mentioned about rash being one of the main side effects of some of these inhibitors, and that's actually true, for instance, in the EGFR space as well. The good thing from the standpoint of the field is that, you know, some of these EGFR inhibitors have been approved for a few decades now, and so clinicians have a lot of experience dealing with rash, understanding it, knowing how to get ahead of it. We used to have a program that we called naporafenib. It's a pan-RAF inhibitor, not to be confused with our pan-RAS. Pan-RAF inhibitor that was being advanced in melanoma. And that molecule did cause a significant amount of rash, but via understanding of the biology and seeing other things that had been done in the past, we instituted what we called primary rash prophylaxis, which is a relatively simple intervention when you think about it. It's topical steroids and it's oral antibiotics given at the right time to hopefully prevent a rash from developing, or at least not a severe rash from developing. And that, if you look at our data pre-primary rash prophylaxis implementation versus after we implemented it, the rates of rash, the rates of discontinuation due to rash, both went down. The relative dose intensity, which is a measure of how much drug does the patient get, if there's any dose reductions or anything like that. The RDI went up in response to us implementing this rash prophylaxis. And so there's a lot of things that we can do. I bring up rash as just one example, but there's a lot of things that we can do to help patients. And I think the field as a whole has made a lot of progress on this front.
I just want to add quickly there. I think David brought a good point there, which is the idea that sometimes wild type plays a significant role in disease, and we should be thoughtful about safety. but in avoiding wild type, you might leave out a lot of efficacy. So in those instances, in the case of EGFR, there is space for innovating around, okay, how can you make RAS better, like the way he was talking about. With amivantamab, there are the Cocoon studies that did something similar, which is how do you, since going after wild type is necessary for efficacy, how do you introduce things that can ameliorate some of the side effects as well?
Got it. That's unfortunately, that's all the time we have. Thank you so much. Thanks for the panelists for all your insight, and thanks for the audience for joining us today.